Hello. We're just going to give everyone a minute to get into the meeting and then we'll get started. Um, Sometimes it just takes a second for everyone to get logged in and we don't want you guys to miss the beginning. I always feel like we should talk about the weather when we're waiting. So how's the weather for you guys?
Cold.
Holds.
Yeah. Yeah, we woke up to 20 degrees this morning.
Ooh,
I know. How about Philadelphia area?
it's been rainy a lot. We're having a lot of rain and cold. But we're going to be in Canada next week and it's warm next week in Canada and there's not going to be snow. We're super bummed.
Oh, that's disappointing. Yeah.
Right?
Snow would be nice. My 8-year-old really
Count
thinks
on
it's gonna
snow.
snow on Christmas just because it's supposed to snow on Christmas, and we're in North Carolina and it's just not going to. So we're working on tempering that. It's gonna be like 60.
Yes. Ooh, yeah, they're not going to get snow.
No, not so much.
All right, let's go ahead and get started. And a few more people I'm sure will join us as we go. Hello, I'm Darla Clayton. I'm the Community Relations Specialist for the Alliance to Cure Cavernous Malformation. And I have the CCM2 variant, and so do both of my kids. So unfortunately, I'm more familiar with CCM than I ever would have liked to be. And I'm excited to welcome Doctors Connie Lee and Amy Akers to talk to us tonight. I'm sure most of you know Connie Lee. She is the Chief Executive Officer and founding president of the Alliance to Cure Cavernous Malformation. Connie is a licensed clinical psychologist and the mother of a daughter with multiple cavernous malformations. Connie's story with cavernous malformation began in January of 2000 when her 4-month-old daughter Julia received brain surgery for a major hemorrhage and hydrocephalus caused by a cavernous malformation. Dr. Lee's passion and drive to help people affected by cavernous malformation led her to start the Alliance in 2002. We're super excited to have an update from Connie tonight. And Amy Akers has been the Chief Scientific Officer for the Alliance since 2009. Prior to that, Dr. Akers began her cavernous malformation research career at Duke University with Dr. Doug Marchuk's lab. While in Dr. Marchuk's lab, Dr. Akers focused on developing mouse models for cavernous malformation and investigating the molecular nature of human brain lesion development. We're really grateful to have Dr. Akers joining us tonight. Before we get started, a couple of quick housekeeping notes. We will have time at the end of this meeting for a Q&A session. And we would really ask you to please use the Q&A feature in the webinar rather than put questions in the chat. If you put them in the chat, there's a good chance we'll miss them. So please try to use that Q&A feature. I'll be checking the chat throughout as much as I can. So if you're having trouble finding the Q&A feature or have questions, and with that regard, pop those into the chat. And then at the end of the meeting, we will be having our weekly support group, which kicks off at 8 o'clock. So I will drop the link for that into the chat a couple of times over the course of this meeting. So without further ado, I will pass it off to Connie and Amy.
Thanks, Connie. I'm excited to be here and to be able to share some of the things that happened last year and that are coming up in the coming year. I'm going to share my screen so that I can follow along with my own slides. There we go. So the title of my talk tonight is Moving Audaciously into 2024. And I hope by the end of the talk that you will agree that we are indeed moving audaciously. It is an exciting time for the Alliance. And just to get you up to speed with what happened just in the last year, First off, this talk is going to focus on research. We do so many things that I can't fit all of them into an hour. This hour is going to be dedicated to the things that we're doing on the research side of the organization. So what we did last year is we completed enrollment of our very, very first industry-sponsored trial, the REC 994 phase 2 trial, and we completed it ahead of schedule. So by next summer, this coming summer, 2024, we're going to have some sense of whether REC 994 is a medicine that can be continued into the next trial because it's safe and possibly even has an effect on the lesions. We completed the creation, and when I say we, I mean you, and this too with the REC 994, we, you and me, completed the creation of the CCM Health Index. Over 500 of you participated in that. And that is the patient-reported outcome survey that's going to be used in clinical trials and is used in other research. We organized a science meeting this year that was just amazing. We heard about progress in focused ultrasound research where mice had lesions stop in their tracks or did not develop new lesions once they had received focused ultrasound. We heard about the beginnings of gene therapy research that's happening at Duke and at the University of Chicago. And we learned about organoids, which is a new way to try to screen medicines. And it's not ready for prime time yet, but it's in development, and it's a really exciting way for us to have an idea of whether or not a medicine is going to work. We funded nanoparticle research this year, and that is a delivery system for medicines that would target specific, hopefully specifically the CCM lesions, but even if it were just specifically brain vasculature, that would be a big improvement if medicines didn't have to be absorbed systemically, causing all kinds of extra side effects. We were asked by another drug company, Ovid, to become partners with them to help them figure out the best way to bring their ROCK2 inhibitor to market, or at least to treatment trials. And so we've been working pretty closely with them. And the fact that they came to us is really indicative of the stage that we're at. We now are viewed as the experts, and pharmaceutical companies see us as really the best resource that they have for moving their trials forward. And then we extensively surveyed the patient community, you all, over the course of the spring to get a sense of what is important, what are your research priorities, what are the things that you would be willing to tolerate in a treatment in exchange for having some significant benefit. So those results were conveyed to the research community, and we're going to be working with them more over the course of the year. And I'm not going to go through this whole thing because I have a lot more to say tonight, but this is the pipeline of treatments as it stands. It's December now. It hasn't changed any since I first created this slide in October. We have many treatments that are being investigated and explored as possibilities for CCM, and we have a good number of them that have already moved into phase 2 now. We have 3 of them in phase 2, and we have 2 more that will be moving into phase 2 within the next year, probably within the next 12 months. So it's just an exciting time as we watch these things progress and these lines get longer and longer. It's because you are participating. That's why this is happening. Yes, we're doing a lot behind the scenes, but it requires every member of this community in one way or another to make these lines move. So what am I going to talk about tonight? Mostly we're going to be talking about, Amy and I are going to be talking about what our plans are for the coming year. But in order to do that, I want to contextualize what we're planning, because many of you have not been with us for the last 21 years, the way I have been, or the last 14, the way Amy has been, or somewhere in between there for as long as Darla has been. So we have been very important in research in the CCM world for a long time. And I just want to roll right through the last 20 years so that you understand where we are now. And then we're going to talk about preclinical models of CCM. So that's before medicines enter people. And then finally, we're going to talk about what the plans are for 2024. So history of our CCM research support. way back in the day, back in 2005, when I was a younger woman, we held our very first international CCM scientific meeting. Amy was there. She was a child at that point. We brought together researchers from, at that point, from just a couple of continents. At this point, we have been bringing researchers together. We have our 20th anniversary this year, and they're coming from 5 continents. And it is the only meeting in the world focused on CCM and has led to tremendous growth in the field, new collaborations, lots of very, very important discussions and data are presented in those meetings. In 2006, we established our CCM Biobank, where we've been collecting tissue and DNA samples. And in the last year, we have expanded and moved it to Texas Heart Institute, where we're now collecting plasma samples and pretty much anything else that is of value to research. Our patient registry started in 2011, and we enrolled over 3,000 patients. We've re-upped it and again moved it to a new platform this year to make it more scientifically rigorous. And so what we're asking all of you to do is, if you are a patient, please re-register if you haven't done so in the last year. Re-register at ccmregistry.org so that we can make this new, more robust registry as important to the field as our original one was. We went on to begin a free genetic testing program to help us prepare for trials so that our patients could already know what their genetic mutation is if they have the familial form before they enroll in a trial. We created an infrastructure of centers that can host trials, and they're offering good care, good multidisciplinary integrated care to our patients, but they also have the potential to host clinical trials in each of the centers of excellence. And then we've engaged in extensive outreach programs. Part of our call is to get patients into trials. That is such an important part of the role that we play. And the need now is for everyone to participate. So we're going to need international trials, and developing international chapters has been a significant part of what we do. We have 21 of them. We have 21 other countries have their own patient organizations as a result of us, and then their grandchildren now of us that are out there working in the field in their countries. And then we have special projects, Breaking Barriers for Black Health Empowerment, the Baca Family Historical Project, and CCM3 Action, all of which have been active at some point in our history to bring in specific groups who are needed in clinical trials. We do direct research and support industry trials. So we organized 2 major meetings with FDA that shifted the way FDA thinks about this disease. The first was with our researchers and industry partners, called a Critical Path Innovation Meeting. The second was just with patients. Only patients came to FDA and shared their stories, and that was probably the more impactful in terms of helping FDA understand that, yes, people have hemorrhages with this disease, but there's a whole lot more to it too. We're members of a 15-year natural history study called the Brain Vascular Malformations Consortium. And then we directly support industry trials by bringing companies in, and that is with BioAxone, a company that has developed a rho kinase inhibitor that's now being brought to trials by a company called Neurelis. We were the ones who brought them in. And we also consult with every company on what an appropriate clinical trial design would be for their particular drug, what sites could be available. We help introduce them to the potential directors of those sites. We recruit the patients, and we educate the trial personnel, both the company and at the clinical research or contract research organizations that they use at the sites. And then, of course, we were instrumental in developing the CCM Health Index. Last slide about the past. We began funding research 2 years ago. So we have now offered grants in focused ultrasound, gene therapy, and in microRNA technology. We support targeted treatment delivery through nanoparticles right now. That work is starting and ongoing at the University of Pennsylvania. Our funding has brought about some pretty significant results, particularly in that focused ultrasound project. We participate in international clinical trial efforts. We are on the clinical advisory boards of pretty much every consortium that is putting together a plan to get funding. And then we connect drug companies who are developing potential CCM treatments with right now to academic partners who have the mouse models that are needed to test the medicines. And this is where I am going to turn things over to Amy, because I think this is where I'm turning them over. Yes, because what we're going to talk about now is what preclinical testing and what preclinical models are out there. What needs to happen for a drug company to decide that a medicine is worth trying in our patients? Amy, take it away.
Hi. Thank you for listening to me tonight. I have my partner Rooster here. I'm sorry, he's probably going to hang with me for my whole few slides here. So I want to talk about preclinical models a little bit. And what is a preclinical model? So preclinical models are basically systems of analysis that aren't a human. It's a way for us to move between a test tube in a lab to get eventually to human clinical trials. So the first model system to talk about are cell models. And what you're looking at here on this slide, there's two pictures of it's basically a plate of human vascular cells, so human endothelial cells. The one on the left is a normal plate of cells. The blue dots are the nuclei in the center of the cell. And then you can see the borders of the cells. They're kind of circular-ish, but those are what normal vascular cells look like. They're very tightly connected to one another. There's a structure to them. But then if you mutate those cells, if you knock out or completely get rid of the function of one of the CCM genes, in this case here, CCM2, there's loss of function of CCM2. So that's not working anymore. And then there's a phenotype that results. So what you're seeing in these cells are those green stripes that come up all over the place. And it kind of looks like someone scribbled on the cell. And those are actin stress fibers. And the point of this is that losing the function of the CCM2 gene causes an effect in the cells. So we can test these cells with different types of drug molecules and see, can we fix this effect? There's also an effect of the tight junctions. So think about a blood vessel cells. The blood vessel cells need to be very, very tightly connected to one another, or else the blood will leak through. Well, in CCM lesions, there's leakage that happens. So there's actually a disconnection between the neighboring cells. So again, we're looking to see, can molecules, compounds, drugs that we might be interested in testing for CCM patients, can they actually rescue these effects? So cell models are fabulous ways for us to kind of screen through a lot of different molecules and say, is there an effect, like I said before, between the test tube and moving to human patients? So we're really interested in testing a bunch of compounds that are already out there. And when I say out there, I mean, these are compounds that are approved drugs for other indications, which means that they've been through the testing process, they're safe for human consumption, and they play a part in the biology that we understand, and drugs that haven't been tested elsewhere in other CCM labs before. So Alliance Cure is really interested in testing this pool of drugs that we think might be potentially interesting for CCM patients down the road. We have the support of our scientific advisory board, and in terms of the molecules we're interested in, and both in terms of the strategy that we're going to be talking about now. So I just said a lot of things. Let me take a look at this slide and see what I missed. We talked about the mutations. We talked about drugs. Endothelial cells are vascular cells. We can also test multiple drugs at multiple dosages again. So cell models are wonderful because you can do a lot at one time, and you can kind of bang through a whole bunch of drugs in different doses and different combinations and see if there's an effect, and then only test those with the most promising results. And once you get to promising results, we want to move up the ladder a little bit. And down the road, Connie mentioned organoids earlier. Organoids are something that may be of use to us as an intermediary, again, between the cell lines and humans. But for now, our next step is to move into mice. So Connie, if you can go to the next slide. So mouse models, what we want to do is create a mouse that has CCM lesions. And how do you do this? Well, there's a number of ways to do it. What we're going to talk about today is we want to create a conditional gene knockout animal. So what does this mean? So conditional means we control the timing and location of the gene knockout. And gene knockout means getting rid of the function of one of the CCM genes. So there's a bunch of genetic tricks and breeding strategies involved in the mice. So what we want to start with is create create a mouse that has a CCM gene of interest. Say we're talking about CCM1, that's the one we're going to be working with. So you want to create a CCM1 allele, so a CCM1 a gene, one of the two CCM genes, and put flock sites on both sides of it. So flock sites are basically recognition sequences that will flank the CCM1 gene. So there's a bunch of genetic tricks that you can do to make that happen. And I'll show you a picture on the next slide. But for right now, just picture it in your head. You have the gene, and then on both sides, you have the flock sequences. And then in multiple rounds of breeding, you end up with those flock sequences on both copies of the CCM gene, CCM1. And then we're going to breed in another gene called Cree. So Cree is not a mouse gene. It's not a human gene. It's actually a viral gene or bacterial gene, excuse me, that acts as a pair of scissors, which you'll see on the next slide. It looks like a pair of scissors. So it is the driver that actually recognizes those flock sites, and you can control the expression of Cree. You can control where it turns on and when it turns on to specifically knock out or get rid of the function of one of those CCM genes. And that control is done through step 3 here in red, which is actually an injection of a drug called tamoxifen, which the Cree gene is responsive to. Tamoxifen turns on that Cree gene and cuts out the gene that you're interested in. So let's look at the next slide of a picture of this, and hopefully it'll make a little bit more sense than all my words. So, excuse me. So again, what we're looking to do is make a tissue-specific conditional knockout gene. Tissue-specific is important because if you knock out CCM1 or CCM2 or CCM3 in every cell of the body, both copies of that have no function of it, animals don't live. They just can't happen. So what we want to do is actually just knock it out specifically in the brain endothelial cells, so just the blood vessel cells of the brain. So we start in blue up top here on the left, the CCM gene. You have those flock sites on both sides. That's done through genetic tricks in the lab. And then you cross that mouse to a mouse that has the Cree is in green. So Cree is the gene of interest that makes the scissors to cut the CCM gene out. And the Cree is actually controlled by what's written up here as a BREC promoter. So it's a brain endothelial cell promoter. So promoter is the control region of the Cree gene. So what this means is that the Cree gene will only turn on within the brain endothelial cells of our experimental mice. So there's a lot of words I'm saying here. At the end of the day, we do a lot of genetic and breeding tricks to create this gray mouse on the left that has both copies of the CCM gene with flock sites on it, and it's positive for this Cree gene, Cree scissors, which will only turn on within the brain endothelial cells. So you give that mouse a shot of the tamoxifen at the time we want to do it, and the scissors come out, i.e. the Cree gene turns on, targets those flock sites, the red triangles, and cuts out, deletes, gets rid of, ruins the function of the CCM gene. And then these mice that we're interested in develop CCM lesions. And then the mice are a fantastic tool. They develop lesions that then we can assess them for all kinds of things that we couldn't test in the cells themselves. So cells are awesome because you can look at the structure, you can look at how they align next to each other, but you can't actually look at the lesions. Thank you, Connie. She read my mind. So once you get into the mouse models, the mouse really do recapitulate the human situation much, much better than the cells do. They develop these multi-cavernous lesions throughout the brain, and they're just a wonderful way that we could track the lesions. So we could ask questions about how do they develop? How do they grow? Can we stop them from developing or growing? Can we actually shrink them? Can we change their hemorrhage rates? There's just so many more questions that you can ask in the mice related to the actual outcome of the lesions as opposed to the very fundamental biology. And the outcome of the lesions, I think, is more similar to what human patients experience. So mouse models, the punchline down here is that mouse models are how we decide whether a drug should be tested in patients. They're really the biggest go in between to go to the next step in doing human clinical trials. What didn't I say?
Well, I think what we have there in the large box about mouse care is something very important to mention. Animals are used only when there is no other choice. So in this case, we use mice because mice are right now the only way that we can figure out whether or not a drug has the potential to be effective in a human system. If it's effective in a mouse system, then we skip the other animals in between on the evolutionary line. We just go from mouse to human. We are not able at this point in the cells to get enough information to to justify the major financial investment that is made in a clinical drug trial. Clinical drug trials are frequently millions and millions of dollars. And so there needs to be some sense that there is efficacy, that these drugs have an effect on lesions. And that's where the mice come in. We can share that within laboratory settings, mice are treated as humanely as possible. There are regulations in place nationally. There are institutional committees that set standards for the way the mice are to be treated, or any of the animals that are participate in university research or in research in private settings. And institutions can get external accreditation if they choose to. So if you have any questions about mice, ask Amy, because I won't be able to answer it, but she's worked in vivariums before. On our staff, also Holly Bly, whom some of you know, has worked in a vivarium. And Jeanbo has worked. We have 3 members of our staff that have experience in this type of setting and can answer questions. So what is happening now in terms of mouse research with CCM? We have some challenges, and that is where we are starting to look for some solutions for next year. The current challenge is that all mice that are used for CCM research are housed in academic labs. There is not a private setting right now that has living, breathing mice that can be used for research in testing treatments. We know this. We assembled a consortium of 13 of the 15 labs that have mice. We came to understand what kinds of mice they have, how they have altered them in order to create the genetics that the mice wind up with. And also we have a sense of what's been tested, what drugs have been tested in these mice. What we also know is that academic labs, what they work for are journal papers. That these cutting edge research papers do not involve factory-like, production-like testing of treatments. That is not publishable. It's also not fundable by the National Institutes of Health. So for them, for academic labs, it doesn't make sense for them to just take their mice and roll through, or take their cell lines and roll through a whole bunch of treatments to see if they get any hits out of it. There's really nothing that of value in that for them, honestly. And they also, I put here that they put surcharges on research funding. If you are to give a grant to an academic institution, for the most part, there's also a requirement that you add another 20, 30, 40, sometimes as much as 60% on top in order to pay for indirect costs, which is like the light bill, and the things that keep the institution open. They're important, but they're not going directly to the staff that's doing the research or the research itself. Academics have limited cages. This is a huge thing. Each institution only has so many cages for housing experimental mice, and each lab is allocated a certain number of those cages. So the labs keep their cages full of mice that are being used for their experiments. That is pretty much all they have room for. The animals that they house are already spoken for the experiments that they're doing. So that's our challenge, is getting in. And what we found, as I'm going to skip to the middle one here, is as we were trying to help an industry partner find a place to test their medicines in mice to see whether or not the medicine would have an effect on lesions, we had a huge problem finding an academic lab that had the capacity to take the project, even though they were offering to pay. This is not work that they expected a lab to do for free. The labs just already had things going on, and there was, we've encountered, as I put here, up to an 8-month delay in getting things moving, which is a deterrent for other industry partners. It's not just bad for this one experiment. It's a problem for everybody who wants to come in. And makes it so that other industry partners will likely say, hey, this might not be where we want to go if we can find somewhere else a different disease that has access immediately, or access within a shorter time frame. The other thing that's happened is that as a result of our accelerating cures work, so the work that we've been doing for the last 2 years, Jean-Beau is not on this call, but he has been a critical part of helping to identify at least 20 potential treatments that have not yet been tested. And these are drugs that we don't know. I mean, they may turn out to be the cure, they may turn out to be nothing, but there's nowhere to do this yet. And they are repurposed drugs. They're all drugs that are already out there. Some of them are already approved for other conditions. Others are already in clinical trials for other diseases. So at this moment, we have to find a solution to make everything happen faster, to not be at the mercy of the tiny amount of space that academic labs have for us, and to not be sending industry partners away disappointed because they couldn't get access in a way that allows them to test what they've got. So we have an opportunity here. And so, as I say, production level treatment testing can give us more options. And the other thing it can give us is the opportunity to test drug combinations that wouldn't be tested otherwise. A drug company is not going to test its drug in combination with another drug if it hasn't yet gone on the market and, done blockbuster stuff. We can do it may be possible for us to start doing that. So we are moving audaciously into 2024 by testing treatments ourselves. We are going to become basically researchers. We're taking on our own direct research program. How are we doing this? Well, we're doing it with partners. I don't have a lab in my kitchen. I'm not doing that. We are partnering with academics, and we're partnering with something called a scientific research organization to carry out the screenings that are needed to determine whether or not there are drugs that we're missing that might have an incredible impact. So our steps here are to contract with an academic lab who can screen in the cell line models that Amy was talking about. And this is something that they do have the capacity, the academics have the capacity to do. We have a specific lab in mind. They will be using human and mouse CCM1 and CCM3 cell lines to narrow the list. We'll take our 20 and see which are the absolute best candidates. And then once we have the absolute best candidates, we do need to take it the next step, which is to go ahead and test them in mice. We are working with a scientific research organization to bring our mice from breeding mice from academic labs to the scientific research organization, which is a freestanding independent laboratory that specializes in preclinical experiments, but that we can direct, and we can tell them what we need to have done, and they can go ahead and carry it out. They're contractors. We will bring in mice from our academic labs with appropriate permissions, and the scientific research organization will conduct the experiments that we design and analyze the results. And so over the course of the next year, our plan, and I'll share that on the next slide a little bit, but our plan is to screen cells, screen drugs in cell lines, and then grow a colony, bring in breeding mice in order to create a colony that we can use to have a sustainable testing program that tests the best candidates from this screening and then from further screenings as new opportunities come along. It's also a place where industry can come and say, hey, we have this idea, we've got this compound, we think that it might have some potential. We can go ahead and do some cell line, well, they will have already done cell line screenings, honestly. That's something that they can do. They can come to us if they are ready to take it the next step and see whether or not their medicines can work in a mouse model. So this combination of working with academic labs and a scientific research organization and us allows efficiency. It allows us to bypass a lot of the bureaucratic hurdles that we face when we're dealing with academia, and gives us some control and certainly a lot more speed than what we would have had if we didn't do this ourselves. So last slide. What does it look like in 2024? We're expecting to pre-screen in cell lines 20 compounds. We expect to have a mouse colony fully up and running by the end of the year, hopefully by the fall, and to have tested the top 2 drug candidates in the mouse models. And then in the following year, and I said fiscal year, I should have gotten rid of that, as calendar year, 2025, we expect to test the next 8 to 12 candidates, and to facilitate anything that comes our way from industry. This is not cheap. This is a big price tag for us. It is cheap relative to what it would cost to do it outside of this model, but it's not a small amount of money for our organization. So in this calendar year, we expect that the cost of those items that I put up there for 2024 will be about $176,000. And then in 2025, as we ramp up testing, the cost will probably run around $230,000 for that year. What we hope to get out of it are treatments. That is the purpose of this. Treatments that are far enough along that a drug company can look at our mouse data and say, yeah, this makes sense for us to take this, or that we can bring it to NIH or FDA or the Department of Defense, who, believe it or not, funds drug studies, and say, hey, this is something that looks so promising. Please consider whether or not you might be willing to fund this trial. So that, in essence, is what our program is looking like. And I think at this point, I'm going to stop sharing and see what questions we can answer for you all.
All right. We have a couple questions in the Q&A. Thank you, Connie and Amy. That was really interesting. And as always, as exactly what I expected it to be, super interesting. We have a question about AI. How might AI contribute to advancements in research, diagnosis, and treatment in the coming years? And is it different between the 3 genetic forms and sporadic?
Is AI different? Is that
I think the question is, would the contributions be different between?
Amy, do you want to start and then I'll pick it up?
I don't know a lot about AI, so I'm certainly not going to speak as though I'm an expert, but in terms of research, I mean, it can point us to new areas. We've chosen a list of 20 drugs that we're really interested in based on what we know about the biology. So if AI can surf the web, for lack of a much better term, and point us in new directions that we hadn't thought of before, or make new connections that we hadn't thought of, I certainly think that's an interesting way to go. And would it be different between the 3 genetic forms? I don't think so, because we're learning an awful lot about all 3, and we're starting to understand, not perfectly, but we're very much starting to understand the biology of all 3 genetic forms and the sporadic form. So I wouldn't think AI would be better for any one of them.
And AI has already been used. Recursion's drug REC-994 was identified using AI visual analysis of the pictures that we showed you on the cells that are abnormal versus normal come from the University of Utah, where Chris Gibson was. He is the founder of Recursion. Those were the first experiments that he did, where basically machine learning identified which cells were rescued or more abnormal than others. We also have AI that's being used in the way that Amy said, which is to really dig deep down into the literature. And we benefit already from that. We have some sense of what is out there from AI. And then finally, the National Institutes of Health has developed a tool that we have access to that uses AI to connect diseases with all of the different biological pathways that are involved with those diseases, and then identify all of the different drugs that are supposed to affect those pathways. So we have looked through the data that NIH has that comes out of this tool. It's not that NIH has generated the data. Their tool generates this data to see if there are ideas there that make sense, that fit our criteria of being something that can be brought to patients. I got to tell you, I mean, like there's some weird stuff on there. They have like strychnine and some poisons. Yes, poisons will probably make a difference in the disease, but that's not something that we want to give our families. So we have to go through. AI is great, but there's some filtering that of reality that has to happen in there.
Very good. Thank you. Um, this is a question, I'm assuming from one of our international folks. All these tests and these research, is it something that will be used internationally? For example, if you find a medicine or treatment that can treat or cure CCMs, can that research be international or is it just going to be used in the US?
No, it's international. It may not be initially international, but there are enough mechanisms in place so that anything that's discovered in the US goes probably first to Europe and then Australia and New Zealand, and then it starts to spread throughout the world. And in fact, some of the trials now are being conducted in Asia. So it would not be, it probably would not take long. And if the other thing is that there are companies or organizations that where that's their purpose is to help people in a country where a drug is not approved get access to it through usually through the company that is producing the drug.
Thank you. All right, this person is asking if there's been some advancements with focused ultrasound to test potential drugs for CCM.
I was okay. So one of the thrills of my last month was that I was able to go to the dissertation defense of Delaney Fisher at the University of Virginia, who has been doing the bulk of the focused ultrasound work. Now, has there been advancement? Watch her video from the patient conference that we had over the summer, and you will be amazed and astounded by the progress that's there. She explains how using focused ultrasound along with microbubbles, which are basically the little puffs of gas, in mouse models of the disease appeared to stop the lesions that were on which the focused ultrasound was focused from growing anymore. It stopped them in their tracks. And then in mice that had not yet developed lesions, so in really baby mice, doing the same thing in the areas that had received focused ultrasound, no lesions have developed, only in the areas that had not received focused ultrasound. So what does this mean? Why does this happen? There's a lot of work that's still left to be done. And that is exciting, and it's going to, as a result of the work that she did and this, they were able to get a $3 million grant from NIH that has now started funding the next stages, the next very exciting stages. And then finally, focused ultrasound also is a potential way to do targeted delivery of drugs. It is possible to take those microbubbles and hang drugs off of them and open the blood-brain barrier with the focused ultrasound and then deliver the drugs. They're not that far with that yet. And the idea is that that's probably what they would use, not so much with pills, things that you can swallow as pills, but more for things that are larger molecules, antibodies, gene therapies, et cetera, et cetera.
Thank you. All right. Is it possible that cures can be discovered for certain forms of CCM, but not other forms?
Yeah.
So for instance, might work for sporadic and not familial.
Yeah, and it is, and it's not, it's a biology question. And Amy, did you want to
No, go ahead, you're talking, go for it.
Okay. All right. So I mean, there are some differences in biology, and and Amy, you can correct me if I'm wrong here. I don't think the difference so much is sporadic and familial as CCM1, 2, and sporadic versus CCM3.
Mhm.
I think CCM3 is the biggest challenge that we have. Sporadic CCM1 and CCM2 will probably find their cure. We will find their cure sooner rather than later. And then CCM3 will get some relief from that same cure, very likely, but may need something else on top in order to have a full cure.
And the biology underneath all that is that CCM1, 2, and 3 act together in a complex. And even in sporadic cases where there's not an inherited mutation, there's still mutations within the endothelial cells that disrupt the function of the CCM1, 2, or 3 genes. So if we can find a treatment that focuses on 2 or 1 or 2 and 3 to a certain extent, it will affect all the familial forms and the sporadic form, but CCM3 has extra functions. So those extra functions may need an extra treatment.
Yeah, but and when we talk about this, though, what I think what Amy and I are both have in our minds are drug treatments. When we talk about mechanical treatments like focused ultrasound, there is no reason that that will not work across the board for everyone in the same way.
Agreed. Yes.
Great. So just to carry on from there, do you expect treatments or cures? Which one's more likely to come up? How's this going to work?
We are working to have treatments on our way to a cure, and we certainly expect both. And they may not come from the same directions. Again, you know, as we're talking about focused ultrasound and the serendipity that happened of discovering that that tool could make a huge difference in mouse brains. We don't know anything about humans' brains, but in mouse brains, it makes a big difference. We may find that while focused ultrasound is making its way through, that there are better treatments that come ahead of that. Or while gene therapy is working its way through, that we will have better treatments in the form of other medicines. And that's what we can't do enough things at the same time. There just is, the more things that we can do at the same time, we don't need to stay focused at this point on just picking one winner or two winners. We need to try everything, and then we will get there. We will get to both better treatments and to the cure.
Got it. All right, let's switch gears and talk about financial stuff for a second. We have a question asking about how donations and matching help fund research and the mouse colony in particular.
We have made the choice to have all of the donations in the last part of the year go specifically to funding this effort. So there's nothing else in our programming or even in our research portfolio that we're going to be using those funds for, aside from working on developing this program of preclinical testing. So if that helps, so please take out your checkbooks tonight, and we can put our address up, or there's always the donate button on the website. We're happy for you to do that. And the matching gifts certainly help, and they help motivate folks who might not otherwise be donating to go ahead and make that gift.
Yeah, absolutely. Next question is, do we have any government grants for research?
We do not, but what we are learning is that with this program, there is much more of a potential for us to get them. This is research that we're doing that is not that different from what is happening in academic labs, so we can compete in the same sphere as the academic labs. Amy, did you want to kick in there?
I don't know what else to say. We can compete in the same sphere as academic labs. And importantly, we're competing for the same dollar pool, but we're not competing for the same outcome pool. Our outcome in the academic labs, you know, the old phrase is publish or perish. And really the currency in academia is publications, and publications lead to grants. And it's a circle that goes round and round. So you keep doing your research. We can compete for that same dollar amount, but it'd be nice to publish. It'd be a wonderful thing. Our goal is really to get treatments into clinical trials. So I think the goals are different, but we can still work together and still partner with our academics, not in a competitive way, but in a way that elevates all of our work and brings treatments faster.
Yeah. And we're also not, there are different ways that things can get funded. One of them is through NIH grants or FDA grants. Another way is through direct appropriations. And so that's another pathway for us to begin mining more and more. Other nonprofit organizations that are patient advocacy, patient research foundations are getting some results in the last few years and getting appropriations. So not yet, but hopefully soon.
Great. Switching back to the preclinical testing, how accurately does the chosen mouse model in cavernous malformation studies replicate the human condition?
So the chosen model is the best one we have, and that's why we chose it. There's been many renditions of CCM animal models. So I said somewhere in my presentation that if you were to knock out both copies of any CCMG, 1, 2, or 3, there would be no progeny that lives. No mouse can live with both mutant copies in every cell of the body. So we've always had to work with different breeding strategies and tools and tricks to create a model. The original models had one completely mutant copy and a wild type copy. So wild type is non-mutant. That's genetic speak for non-mutant. So completely mutant, non-functioning, and a wild type. So that mouse was basically fine until over time it acquired mutations that may at some point in time knock out that second copy. So somewhere within the mouse, hopefully within the brain blood vessel cells, they would acquire 2 mutations within the same cell, seed the formation of the lesion. As you can imagine, this is a very kind of random stochastic event that doesn't happen all that often. So research would breed in different genes that cause mutations to happen more regularly. Sometimes it'd be a cancer type of gene that just simply causes increased mutation frequency. These worked, but they weren't super robust models. So now with the new conditional models, they're awesome because you can control the timing. You can control the location. So by using that Cree driver, the scissors that I was talking about before, you can specifically turn that on at a time point after you inject the tamoxifen. So tamoxifen is the drug that you inject into the mouse. If you inject it at a very early time point, like when the mouse is 1, 2, 3 days old, that tamoxifen works super fast and you get tons and tons of lesions, specifically within the hindbrain of the hind part of the brain, which is interesting for answering some questions about lesion development. But if you inject it at a later point in time, after kind of that early angiogenic period where the brain and the blood vessel cells have already formed some, then you get lesions which are more distributed evenly throughout the brain, but you also get these really mature multi-cavernous lesions that look an awful lot like human lesions, just on a smaller scale. So it is the best that we have. It's the best model that's out there right now. It's pretty darn good.
Awesome, thank you. We have a whole bunch of comments in the chat and also in the Q&A, just generally saying that you guys are awesome and thanking you and etc. for being so fantastic. So I just wanted to make sure that you all heard that. You guys are great and we love you. And I think we have time for probably these last 2 questions, and that's likely about all we have time for because we do have support group right afterwards, and I have to start that meeting, so that will be at 8 o'clock. So we have a question about a person whose husband was just recently diagnosed, and it's all very new to them, and they're wondering if there's any medications or anything that they can do right now to help with symptoms besides they're taking something, it looks like maybe for pain and seizures.
Yeah, I think what we have been encouraging folks to do, and I think there's enough research evidence to support that, is to make sure that your vitamin D levels are in the normal range. You don't have to have extremely high vitamin D levels, but your doctor can do a blood test and determine whether or not you're low on vitamin D. And if you are, supplement, and your doctor will tell you what the appropriate level of supplementation is. And I want to say, do this through your doctor because you may need way more vitamin D than what you can get over the counter, and that would help things get better faster. And then the other things that we encourage folks to do are to not eat preservatives in your diet. We have evidence and research, again, this is in mice. We don't have it all worked out in humans yet, but that eating foods that are full of what we call emulsifiers, or that also known as preservatives, can inflame your gut. It reduces the amount of mucus that's in your gut. It can cause an inflammatory response, and that inflammatory response can go ahead and can work on the lesions in your brain. So if you want to keep your lesions quieter, one of the ideas is that you try to eat from the vegetable, fruit, and meat departments rather than from the Twinkie department. It's just one of those things, and we're really sorry about that because I like Twinkies too. But it's better to not do that. And those are the two main things that we can recommend at this point. Amy, do you have any others that you can think of?
Nope, those are the biggies. That's great. Yep.
And genetic testing is important. So if you have multiple lesions, because I think I saw in that comment that they don't know what kind he has. If somebody has multiple lesions that can't be explained by the presence of a developmental venous anomaly, then they qualify for genetic testing. And you can get testing for free through us. So the way to get that is to register in our patient registry, ccmregistry.org, and let us know. There's a question there that says, do you have multiple lesions? Have you had tested, or has a family member had testing? And if not, then let's go ahead and see if we can get you signed up.
Great. All right. This, I think, is just such a fantastic question. I saved it to the end. I think there's lots of things that we can suggest for this, but this person is asking what they can do to help.
Oh my gosh.
I know. It's a great question.
Oh my gosh. Well, who are they then?
It's an anonymous question.
It's an anonymous question. So it depends on what your skill set is, anonymous person. Everyone can do something. And so if it is just spread the word to your family that the work that we're doing is important, or information about the disease itself, or share it with your friends who may not have any exposure to what CCM is. The more people who know about the illness, the more likely people who have it are going to know there's resources. So there's that. There is also on our website a volunteer form that you can fill out. And so if you do have particular skill sets, go ahead, fill that out, hit send, and it will go to Lindsay and Darla and Linda, and they are able to figure out what the best fit might be for you. Obviously, if you have financial resources, and I know that in our community, a lot of people have a lot of medical expenses and don't, but if you happen to be one of the folks who do, a donation is really appreciated. None of this is free. None of it is free. All of the work that we do has some price tag attached to it, and we want to continue to do it and do even more. One of the realities that we have financially is that over the last 3 years, we have been recipients of the Chan Zuckerberg Institute's Rare as One grants. Those were $200,000 a year grants for 3 years, and we benefited greatly from them. They are only for 3 years. At the end of 3 years, they go away. So we received our last check just this past month. Starting next year at the end of 2024, we need to figure out how to make up the difference, and at the same time expand our program so that we can do mice. And we have a lot of thoughts for how to do that, but any support that you offer would certainly be helpful in that regard. Did I answer enough, Darla?
I think you did.
Okay.
You think of anything else, Amy?
No, I think it's covered.
Yeah. Well, okay, sign up for the patient registry.
Sign up for the patient registry and come show up. Just show up. We have things going on all the time from a community perspective. We have support groups. We have gatherings, get-togethers in different places. Just come and be part of our community. It really does make a difference.
Come to family camp. We have family camp.
Come to family camp. Yeah, family camp is going to be this June. That was hard for me to get there. And it's going to be a lot of fun. It's going to be in New York. So if you do need info about that, you can always shoot me an email. We would love to see you there.
And our early registration ends the end of this month. There is still registration after that. It's just a nudge more expensive. But if you want the most discounted price and you think you're going to come, go on our website and get to the registration.
Yeah.
And we'll just put her email in the chat. So if you need it.
If you need it. And I think everyone should have the support group link. I dropped it a couple of times. So I will leave the meeting open just for a minute for everyone to grab that link. And then at 8 o'clock, we're going to be starting that meeting. Thank you guys so much.
Thank you, Darla. Thanks for being wonderful. And thank you, Amy, for everything.
Thank you. It's great to be here. This was fun.
All right. Take care. Have a great holiday for those who are having holidays coming up, and we will talk to you in the new year.
Thanks, Darla. Bye. Bye, Ken.
Thanks. Bye.
All good?
Oh, good.